Hey folks, welcome to your very first notes video for Introductory Chemistry. As you should with all of these notes videos, be sure to write everything down in your notebook that's not already on a cutout, glue the cutouts into your notebook when the information on them pops up in the video, and complete any necessary work on the cutouts as instructed. To get the most out of these notes, you should definitely watch and listen to the video. There's a lot of important stuff that I say, including extra credit opportunities, that you won't find on the text of the slides. In today's lesson, we're going to learn about what chemistry is, what kind of equipment we use in chemistry, and some basic lab safety concepts. Let's get started. First off, what is chemistry? Chemistry is the study of matter. What's matter? Nothing. What's the matter with you? Matter is anything that takes up space and has a mass of its own, which is pretty much everything. The general goal of all science is to better understand the way our world and universe works. When it comes to the specific science of chemistry, we're focused on the different atoms and molecules that make up our world and how they react together. In general, to do science well, you have to be objective and be able to perform repeatable experiments. In other words, scientists should be able to review the data they have available and create their own conclusions solely from that data, not from any other feelings, preconceptions, or opinions of their own. They have to consider all of the data, not just the data that fits their own worldview. They also have to perform experiments or make observations to collect that data. Those experiments have to be able to be repeated by other scientists in order for the data from those experiments to be accepted. If the experiment doesn't give repeatable results, those results should not be used to formulate scientific theories. The whole point of doing science is to answer questions about our world. You may have learned about the scientific method being a linear progression of steps that lead to a conclusion. This is a fine way to think about it when we first learn about it, but this circular map here better illustrates the real-world scientific method. Generally, we start by making an observation about our world. This could be observing some natural phenomenon or the results of some experiment. The next step in the process is to think about that observation and ask yourself why that occurs in the way that it does. This is the big question we're all trying to answer as scientists. Why does this happen? Once you know exactly what question you're trying to find an answer to, you're going to try to predict what that answer might be based on knowledge you already have about that phenomenon. This prediction, or hypothesis, isn't just a guess, it's an educated guess that helps you determine what kind of experiment or test you need to run in order to confirm or refute your idea. So you come up with some experiment to test this idea, gather results from that experiment, and in a perfect world everything works great and you can move on to developing a scientific theory or explanation about why your phenomenon occurs that's backed by scientific data. In the real world, we often do not get the experimental results that we expect, or we might make some new observations while performing the experiment. This may lead us to edit our original hypothesis, throw it out altogether, or start asking new questions. This is where our circular model sometimes gets stuck in a loop, but hopefully we eventually break out of it and end up being able to move on to generating a scientific theory. If you'll remember from 8th grade, scientific theories and scientific laws are very different from each other, but both are generally accepted as truth and are backed by scientific data. Scientific laws are just descriptions of natural phenomena. For example, the law of evolution simply states that over long periods of time, the traits of a species will change and often form new species. The theory of evolution by natural selection uses the process of natural selection to explain why the species evolve over time. A law simply states what is observed, and a theory explains why it happens. You may also remember that laws don't really change because the world tends to keep on working the way that it does, but theories do change as we gain a better understanding of why things happen the way they do. Okay, now that we have the general idea of science down, let's move on to chemistry. There are 5 main branches or fields of chemistry: organic, inorganic, physical, analytical, and biological. Organic chemistry is the study of molecules that are largely made up of carbon atoms. Most medicines and all plastics are made up of largely carbon-based molecules. Inorganic chemistry is the study of molecules that are not made up of carbon atoms. Things like minerals, metals, nonmetals, and semiconductors are made up of inorganic compounds. We study the behavior and changes of matter and how its energy changes in physical chemistry. This might include the study of reaction rates or the steps to chemical reactions. In analytical chemistry, we analyze things, basically. We try to figure out what substances and mixtures are made out of. Analytical chemists are often in charge of quality control, or analyzing water samples, for example, to see what types of pollutants are present. Lastly, biological chemistry, or biochemistry for short, is the study of matter and chemical reactions in living organisms. Biochemistry often overlaps with organic chemistry, as biomolecules are largely also made up of carbon atoms. Analyzing DNA or the effect of certain substances on living organisms are two examples of biochemical studies. Next, let's talk about some glassware and equipment that we'll likely use in this class. First off, a beaker looks like this. We use it to hold liquids and can perform reactions in them, but not ones that bubble or fizz, as the bubbling can cause our chemicals to splatter out of the open mouth of the beaker. We can use beakers to roughly measure volumes of liquids, but they're not very accurate. An Erlenmeyer flask, which has a long neck and a cone-shaped bottom, has the same exact function as a beaker, but due to its special shape, it's better at stopping liquids from escaping in a violent bubbly reaction. A Florence flask is the same as an Erlenmeyer flask, but it has a round bottom instead, and you cannot measure volumes with a Florence flask as there are no volume lines on it. This is a graduated cylinder, and the most often used piece of volumetric glassware that we'll use. They are great at measuring volumes, but you never want to perform a reaction inside of one. They can usually measure up to 1/10 of a milliliter. Due to surface tension and capillary action, the liquid inside of a graduated cylinder will create a curve at its surface. That curve is called the meniscus, and to accurately measure volume, you look for what milliliter marking the bottom of that meniscus touches. A pipette and pipette pump measures volumes very accurately, but usually fairly small amounts of 25 milliliters or less. Burettes are also very good at measuring very accurate amounts of liquids, usually up to 50 milliliters, and these are especially useful when you don't know exactly how much you're going to need, but you need to be able to measure it after you've used it. Test tubes are great for performing small reactions in, or heating things up over a flame, but you can't measure the volume of a liquid with a test tube. Reaction plates have small divots in them that allow you to perform several microscale reactions to test for different properties. A glass stir rod is, you guessed it, used to stir things. A mortar and pestle is used to crush things into a fine powder by pushing down hard on the solid crystals with the pestle in a twisting motion. And a crucible is used to heat up a solid over a flame, either to melt it or dry it out. Here's some of the equipment we'll use. A hot plate is a great way to heat things up. Be careful with them. They get very hot and will melt their own cords if you get them too close. Some hot plates also have a magnetic stir. Magnetic stirrers have a magnet under the hot plate that spins at different speeds based on the knob position. You'd place a magnet in the container you're holding your liquids in, set it on top of the hot plate, turn the stirrer on, and the spinning magnet below will cause the little white magnet on top to spin too, which stirs your mixture. An electronic balance helps us to take the mass of different things. We often use them to measure out masses of chemicals. You always need to place whatever you're trying to measure, especially liquids or powdery solids, in some other container and not directly on the electronic balance itself. We often use a weigh boat for this. An analytical balance is the same as an electronic balance, but it's fancier and measures much more precisely, usually up to the 1,000th or 10,000th of a gram. A test tube holder helps you hold test tubes, especially when you're heating them over a flame and don't want to touch the test tube with your fingers. Test tube rack is a great place to put several test tubes as you perform multiple reactions. You can also place your freshly cleaned test tube upside down on the pigs afterwards so that you can dry them. This is the test tube brush, which you use to clean test tubes and other small pieces of glassware. This is a beaker brush, which you use to clean beakers and other large pieces of glassware. These are beaker tongs, which can be used to grab onto hot beakers so you don't burn your hands. You can also use a silicone glove or hot hands mitt. These are crucible tongs, which are a kind of a catch-all tong that can grab onto a variety of different items. A scoopula is used to scoop out a solid substance from their containers. A spatula or microspatula is also used to scoop out solid substances from their containers, but they do very small amounts at a time. A Bunsen burner produces a flame that you can use to heat things up. When setting these up, you should adjust the amount of gas, which is propane in our case, and air that flows through them so that your flame is just different shades of blue, no yellow or orange. A striker is used to create sparks with a piece of flint so that you can start your Bunsen burner flame. Insulating hot pads are incredibly important when dealing with glassware. Anytime you take a hot piece of glassware off of a hot plate or flame, you'll want to sit it on one of these insulating hot pads. If you don't, the drastic temperature change from the really hot heat source to a really cold lab table can shock the glassware into shattering into bits. And lastly, a ring stand, which can be used with several attachments, helps us hold our equipment in place so we can perform experiments safely. Ring clamps hold clay triangles or pieces of wire gauze, which you use to hold crucibles over a fire. Utility clamps are great for holding just about anything, but especially for test tubes. And burette clamps are used specifically to hold a burette. Okay, last but certainly not least, here are some important safety rules. Regardless of what kind of chemicals we're using, you will almost always need to wear safety goggles during a lab. Eyes are one of the most difficult organs to repair, so protect them. We never eat food or drink beverages in the lab area, which is all of the classroom north of my desk. If there's fire involved in our experiment, make sure you have any long hair pulled back and don't wear clothes with baggy sleeves. For any lab, you'll want to wear closed-toed shoes. That means no sandals. And that's in case glassware breaks or chemicals spill. If any glassware breaks or chemicals spill, let me know immediately by shouting, "Code 1, Code 1." This is better than "Hey, Mr. Kale," because it alerts me that there's an emergency I need to attend to right away. Do not try to pick up broken glass on your own. I'll take care of it for you. Even if you don't think you got any chemicals on your hands, it's best to always wash your hands thoroughly with soap and water after the lab. There's hand soap at each sink available to you. You'd be surprised how much stuff you can get on your hands without realizing. Always wash all of your glassware when you're done with it. I don't expect you to put it back in the cabinets where they came from, but clean it well and with dish soap and hang it up on the drying racks to dry. Lastly, when you pour liquids or solids out of the stock bottle, whatever you take out never goes back in. This is to avoid any contamination for the whole stock bottle. Well, that's all for today, folks. That was a lot of information to cover and will probably be the longest video you get all year. Just make sure you wrote everything down and that you utilize the Quizlet cards and other resources I've given you to help you study. If you have any questions at all, don't hesitate to ask.